virtual event. I'm Peter Daniel, professor and chair of Hofstra's Department of Biology, and I will serve as moderator for our panel of speakers. Our speakers are happy to answer your questions. To ensure a smooth question and answer session, please type your questions into the Zoom chat dialog box, which you can find by hovering over the bottom of your screen. Just click on the chat icon to open the chat box. After the presentation ends, I will select questions and give our presenters a chance to answer. To ensure an enjoyable presentation for everyone, all speakers and video feeds have been muted. And now to introduce our speakers. Carol St. Angelo received her BA in biology from Mississippi University for Women. She completed her PhD in molecular biology at Cornell University, focusing on the replication proteins of influenza virus. She is currently the Department of Biology's laboratory director. Nate Rigel received his BS in microbiology from Penn State University and his PhD in microbiology and immunology from the University of North Carolina. Prior to joining Hofstra, he was a postdoctoral research associate in the Department of Molecular Biology at Princeton University. Nate was working on a variety of bacterial pathogens over the years, including Mycobacterium tuberculosis, the causative agent of TB. With that, I'll turn the presentation over to our speakers, starting with Dr. St. Angelo.
Thank you, Peter. Before we begin, I just wanted to say that enthusiasm about virology isn't the same as enthusiasm about viral illness or pandemic-induced global recession. I mean, I really appreciate what viruses can teach us about biology and about evolution. I appreciate their structure and their genetic economy, but that doesn't minimize the real pain that this particular virus is causing right now for so many people. But since viruses aren't going away, we might just as well learn about them. So let's begin. Viruses are really a unique sphere of existence. Like all living things, viruses have genes and can evolve, but they don't have any metabolism and they self-assemble from proteins. So to have a debate about whether viruses are alive or they aren't alive, it just misses the point. Viruses are their own thing. Sometimes you'll hear people make reference to live virus or live vaccine, but all that really means is the virus is capable of being replicated. And indeed, that sort of passive language is the preferred way to talk about viruses. So rather than say viruses replicate, we say viruses are replicated because there's no agency at all with the virus. Viruses are everywhere. All living things are infected with viruses: bacteria, protozoa, algae, of course, plants and animals, fungi. Everything is infected with a virus everywhere in the biosphere. We now even know that every drop of seawater has 10 million virus particles. And we're really appreciating that the death of virus-infected cells in the ocean is an important part of nutrient turnover. So we're just learning things about viruses all the time. Coronaviruses in particular are RNA viruses. Now, living things have genes of double-stranded DNA. Viruses, depending on the virus, they can have all kinds of genes: double-stranded DNA, single-stranded DNA, double-stranded RNA, and single-stranded RNA. As it happens, a lot of human pathogens are the RNA type of viruses. And of all the RNA viruses, the biggest ones are coronaviruses. Just as a point of comparison, the genes of coronaviruses are about 3 times bigger than the genes of influenza. There aren't a lot of human pathogens or a variety of human pathogens in the coronavirus group, but there are a number of livestock pathogens. Viruses replicate by binding to a protein on the surface of the cell And that basically tricks the cell into bringing it inside. Depending on what the genome of the virus is, there might be a couple of steps, here in the middle. Ultimately, what you're going to get is a lot of protein replication, protein synthesis from the virus, and the cell does all of this. So the ribosome is a cellular structure, and it's going to take care of all the the protein synthesis. And then all these proteins just spontaneously self assemble. And this is one of the sort of really interesting things about viruses is how protein and nucleic acid can just self assemble into a structure that then can survive sometimes prolonged periods outside. And then once the virus is back inside another cell, can spontaneously disassemble again, release the genome, and start a new cycle. The number of virus particles that are released from an infected cell, it depends on the virus. I mean, some cases you can get easily 10,000 virus particles released from one infected cell. Now, that obviously is going to kill the cell immediately. Other infected cells release virus progeny slowly, and then in sort of slow and steady for really years and years. And then in yet other cases like herpes simplex, you've got sort of a sporadic release of virus and then long periods where there's no activity at all. All of the viruses we used to know about, the only viruses we knew about were those that made us sick, because that was the only way we had to know about them. But now with modern sequencing techniques, we realize that there's lots and lots of viruses that infect us that aren't causing any problem of any kind. And that understanding is sort of like what's going on with bacteria. We now know how many bacteria infect all animals and how important they are. I don't know if as many viruses are as helpful as bacteria. There's probably way more examples of helpful bacteria than there are of helpful viruses. But one really intriguing hypothesis is that retroviruses that way back at the beginning of mammalian evolution, before that, as they became part of the genome, because there are a number of retroviruses that are part of our genome, that they were important in the evolution of placenta and so allowed for the internal development of young in mammals. And without that, it wouldn't have evolved. Viruses that do cause disease, though, get most of our attention. So I thought it would be worthwhile just to talk in general about where virus disease comes from and why sometimes there's such a wide presentation in for a given virus for the disease. So there's basically two parts. You've got the virus killing cells, and you have the immune system trying to defend the organism. And sometimes in that defense, there's some pathology. So first of all, you have virus killing cells. So this is just an example. On the left-hand side, you've got nice, healthy cells growing in a dish, and they're all nice and flat, and they look good. And on the right, this is what happens to those same cells when they're infected. And I think in this example, it's West Nile virus. You can see how they round up, and so they're dead. And dead cells are not good for the organism. You don't have that activity. But the other part of pathology from an infection is immunopathology. And I want to be careful not to give the wrong impression, because our immune system is absolutely essential. We'd all have been dead before the age of 2 if we didn't have an immune system. But the immune system, which is this really big, intricate, complex system, we've got innate immunity and then adaptive immunity. One estimate is that about 20% of all of our genes have a role in our immune response. So this is a huge system. So as it's defending us against the constant onslaught of pathogens, it's probably not surprising that every now and then they get a little too exuberant and there's some pathology, whether it's from neutrophils, from excess of interleukins. And this certainly does seem to be the case in COVID-19, that a lot of the pathology is immune pathology. And so I thought, looking at the patterns that you can see, let's sort of generalize a little bit. So on the right, we see rabies. So rabies infects central nervous system. Really, really important organ system there. Kills cells. And so the result is, if you're looking at the intensity of symptoms across the bottom here, that there's a pretty narrow range of presentation. Everyone who is infected with rabies virus has the same terrible outcome. Now, on the left, common cold, whether it's caused by coronavirus, rhinovirus, which is the most common, adenovirus, there's a number of viruses. These are infecting your upper respiratory tract. Now, your upper respiratory tract is much more tolerant of cell death because cell turnover happens all the time. And so, generally speaking, right, you're very mild to no symptoms at all. Doesn't really matter that the virus infects, kills some cells. There's some immune pathology, so it's a broader range of symptoms. But in general, of course, common cold is not a serious infection. And now what I put on top of this is influenza and COVID-19. Influenza, we know a whole lot more about, and you'll see here that the range is really broad because both of these viruses not only infect the upper respiratory tract, they can travel to the lower respiratory tract. And now you've got a much more sensitive tissue, much less tolerant of cell death, much less tolerant of immune pathology. And that loss of oxygen transfer that you get as a pneumonia develops means you're going to damage all these other organs. Add on to that variation in age, in nutritional status, in other infections, comorbidities, and you end up with this huge range of presentation where some people show basically no symptoms at all. Some people show really severe illness. Now, I put a dotted line in over here for COVID-19. We don't really know where this goes. We know that certainly illness is more severe than influenza. It would certainly seem that the case fatality rate is higher than influenza. Once we have more data, we'll know exactly where that line goes. But whether there is disease or there's no disease, really the virus doesn't care. Viruses don't care whether you get sick. The only successful virus is a transmitted virus. And so viruses are successful if they are transmitted. If a virus produces symptoms like sneezing or coughing that gets the virus to the next person, well, so much the better for the virus. That's great. Some viruses get transmitted they don't do anything to get them transmitted. They're sexually transmitted or they're transmitted in blood products. And they're just riding along with behaviors that humans are doing anyway. And they don't do anything to influence transmission. They just are transmitted. We see a really interesting trajectory, though, and I hope you'll find this in a way, that there is a trend in infectious agents from more virulent viruses over time, they evolve to less virulent. And it's not just viruses, it's bacteria as well. We see cholera epidemics can follow this trajectory. Whereas when viruses first enter a population, if they're quite virulent, lots and lots of virus particles are released, but individuals are so sick that they die quickly or they're not out and about and they can only infect people that are really right next to them. And so the R naught, as Nate's going to discuss in a bit, is not necessarily that high. And the trend over time is for the virus or the agent to evolve into something that's less virulent, that maybe sheds less, there's less virus released, but the infected host is up and about and feeling well enough to go about glad handing everybody they come across and thus spreading the virus effectively to more people. So that becomes the more successful virus. And I'm going to end my part of this presentation with a really interesting case study with the pig coronavirus TGEV, which stands for transmissible gastroenteritis virus. So as this is I put the piglet there, it's a pig virus. This virus causes a really severe gastroenteritis in piglets. The pigs either die directly or they get so sick they have to be put down. And this virus has been circulating all over the country, all over the world, but in the United States since the 40s, and was causing really periodically serious losses in livestock operations. And in 1984, a spontaneous deletion just arose in the S gene. So the S gene codes for the S protein, which is the main spike that makes the corona of the coronavirus. And that new S gene or the S protein, that's the protein that binds to the surface of cells. Instead of binding to cells in the gastrointestinal tract, it could only bind to cells in the upper respiratory tract. It caused a much milder disease, and it actually served as a natural vaccine because immunity toward the deleted version protected against TGEV. The virus was very transmissible, so that was successful. And this virus, TGEV, has basically disappeared from the United States. Now, I have to, for full disclosure, there also has been much tighter security in American agricultural organizations for lots of reasons since the 80s. But this is a big part of it, this evolution of spontaneous evolution. And Nate's going to talk a little bit more about that too. And with that, I'm going to stop sharing and let Nate take over. Okay.
Okay.
Okay. Oh, that was smooth.
Yeah. Smooth for now. Let's give this a try. Share my screen. Okay. I think we're off and running. Carol, thank you very much. I think Carol has given us a really nice introduction to what viruses are all about. And so what I'd like to do with my part of the presentation is focus a little more on some of the molecular details of what makes viruses tick. So for starters, I think one of the things that really interests me as a microbiologist in viruses or any microorganism is that it allows us to study some basic fundamentals of biological sciences. There's really no better model organism than a microbe to study the process of evolution. And so what I'd like you all to consider is that coronaviruses have been with us for quite some time. And every moment that a coronavirus has been associated with a human being, they're evolving. And so you can imagine if you look at a population of humans that are infected with coronaviruses, uh, by and large, those infections produce mostly mild symptoms. So coronaviruses are a major cause of the common cold. You get this virus, you have the sniffles. It's annoying for a couple days, but mostly it's things you can manage with over the counter medication. But when coronaviruses first contacted humans, it's hard to know at what point that interaction began and what the symptomology was at that point. As Carol mentioned, a lot of infectious diseases, or at least during outbreaks or epidemics, there's an evolutionary benefit to maybe backing off on causing really profound symptoms or killing your host. If the job of a virus is to continue to propagate, to continue to make more viral particles, then it stands to reason that maintaining the host environment, having a good place to build more viral particles is beneficial. And so that's a really interesting question at just a basic level for any virus or any interaction between a host and a pathogen. So before November 2019, there were at least 6 separate introductions of coronaviruses into humans. And so what this figure illustrates is a nice pictograph of where we think these coronaviruses came from and just how long ago they popped up. Now, obviously, we can't go back in time 800 years to sample viruses from an infection. But what this really represents is the power of molecular sequencing techniques. We can look at nucleic acids, and you can use this molecular clock analysis to figure out where a particular virus might have diverged evolutionarily from viruses that are circulating in current populations. And so if you use this analysis, you can walk back through time and figure out what different coronaviruses look like and possibly where they emerged from throughout human history. And one thing that I want to point out at this stage is notice the chain of events here. We start in animal reservoirs. We move from one animal to another in many cases through an intermediary host. And then from this intermediary host, we get contact or emergence of these viruses in humans. That's absolutely critical for understanding how the outbreak that we're now facing, the COVID-19 outbreak, how that really came about. And the driving principle here, I think, is just one of the wonders of biology. It's evolution. And one of the keys to understanding evolution is understanding what the word mutation means. One of the important features of the coronaviruses, or any virus really, to survive long term is the ability to diversify, the ability to find a new way to adapt to changing conditions. Maybe it's spreading through different populations, or in the case of these coronaviruses, maybe it's spreading between different types of hosts. And for this to occur, usually it involves some type of genetic trickery, including the cropping up of random mutations. Now, usually when you hear the term mutation, you think that's bad, a mutated gene. That gene doesn't have any function anymore. And it's true that in many cases, mutations result in defective viruses. But that's not always the case. Some mutations might have a different impact on the ability of a virus to continue an infectious cycle. Maybe it has no impact, right? And you can imagine that those mutations might be stable over a long period of time. It's, the deleterious types of mutations, the one that I think most people are more used to hearing about. Those types of mutants would likely be eliminated just through the power of selective pressure, through evolution working its magic. But it raises an interesting point because one of the coronaviruses that was first on the scene in 2002 was SARS. And one of the most interesting things about SARS is after it emerged, it very quickly burned itself out. It went away. Now, it just didn't magically go away on its own. But what we now understand, or the way we think about how SARS ran its course, is really through the lens of understanding how mutations work at the molecular level. And so what this figure illustrates is the basic idea of how viruses contact a receptor cell. And so what you see at the top here is a human receptor protein called ACE2, which is what interacts with the spike protein that Carol had mentioned on the surface of the SARS particle. This interaction shown here, panel A to the left here, this is a nice tight interaction. It's a hand in glove, lock and key type of interaction. That strong interaction is likely to lead to a successful infection. What happens what happened with SARS, or what could happen with any virus circulating in a population, is that subtle mutations crop up over time in the spikes of the SARS particles. And so you see in the middle panel here, we have two mutations in the spike protein. Here on the right panel, again, two different mutations in the spike protein, all of which impact the interaction with the host cell receptor and the spike protein itself. And that defective interaction leads to a poor prognosis, at least from the aspect of the viral particle. It would inhibit the ability of these two molecules to be in contact with each other. And that's what happened with SARS. So if you looked at SARS population, the SARS strains that were circulating in 2002, 2003 at the height of the outbreak, you see that there was this very tight interaction between the host receptor and the viral spike protein. Fast forward a few years to SARS particles that were circulating in the population. They now have these mutations, lessening the impact of their ability to interact with the host receptor. So the term or the concept that we just discussed, of course, it's science, there's got to be a term to describe this. The idea of mutations cropping up in a population of viruses over time is known as antigenic drift. So the idea is that an antigen, some surface marker of a virus that's recognized by the immune system, its flavor changes subtly over time. It drifts from its original composition to something new. And that slight variation occurs through mutation. Now, it's a little different from this term called antigenic shift, which I'm going to describe to you next. And that's a case where you have major changes in a virus that's the result of genome mixing. And what's important to understand, particularly with the coronavirus, is what's available for a coronavirus, or what's more likely to occur for a coronavirus, antigenic drifts or antigenic shifts. What you see in this figure is an illustration of this genome mixing idea, where you have different viruses, in this case, it's different influenza viruses infecting either pigs or chickens or humans. What can happen with the influenza virus is that it's got a segmented genome. It comes in 8 separate pieces. And that's a little different because with a coronavirus, you're just dealing with one segment of a genome, one piece altogether. Now imagine a scenario where each of these viruses is able to infect a pig. Now it might not cause disease. The chicken virus or the human flu virus might not cause flu in this pig. But the fact that you have the presence of all 3 influenza strains now circulating in this pig actually leads to this ability of viruses to be packaged with a completely scrambled content. Okay, and so that's what the color coding represents, that the swine virus has this pinkish colored genome, birds are blue, people are green. You can get this genome mixing as you package these viral particles, and the resulting flu, this hybrid molecule now, that's something that could become something that's very deadly, right? And that's why year to year, when you go for your flu shot, there's a different composition of antigens or a different flavoring to the cocktail that you're going to get. It really has to do with the ability of this particular flu virus to change up its surface composition, to change up its genome, relying on this ability to shift its antigenic composition. That's not something that's as likely to occur with coronaviruses, and that really has to do with the basic molecular biology of how the viruses are put together. And so this just really illustrates that point. Again, the idea with antigenic shift is just little bits of mutations that are coming up here. The antigenic drifts are random, the antigenic shifts are the shuffling events, and that's going to lead to these new types of flu particles. Now, in addition to the antigenic shift and the antigenic drift, Some of the intrinsic properties of the influenza virus also subject it to increased rates of mutation. And one of those properties is the fact that influenza is just very sloppy about replicating its genome. So it doesn't really check on the mistakes it could make as it's replicating. And that again can lead to further mutations in the influenza strains. That's really important to understand the composition or the differences in viral particles that might emerge in a population, particularly when you start to talk about how a virus can contact and infect other people. And this slide illustrates the concept of R0, R0, which is a number used by epidemiologists to basically assess the ability of a given virus to infect people. And so there are, in no particular order here, other than in the order of infectiousness, are several different viruses that are fairly well studied, and their calculated R0 value. Now, I'll note that the COVID-19 R0 value, that number isn't as well established yet. It's simply because we're in the midst of the pandemic. We don't really know where the numbers are going to go. But the idea is that some of these viruses are really, really infectious. All right, so if you look at something like measles with an R0 of 9, that it's something that can spread very rapidly throughout a population. And you contrast that to these different coronaviruses like COVID-19 or SARS or even MERS, which is a different coronavirus. So the R0 value really is a function of the properties, the intrinsic properties of the virus and how it can contact or invade a host cell. This slide breaks out the 3 major coronaviruses and does a comparison with flu virus. So again, COVID-19, SARS, and MERS are all coronaviruses. And here you can see it's this one large genome packaged inside the viral particle versus flu, which is made of this segmented genome inside. And the relative R0 values are shown here. And I think most of you are aware of the relative rates of hospitalization or the infectious rates of these various different viruses. So something like flu, we deal with that every year. There are upwards of 1 billion people that get infected with flu each year. Obviously, the number's not out for COVID-19, but in the case of these other 2 coronaviruses, SARS and MERS, the numbers are far reduced in comparison to influenza. And so what will be keenly of interest for epidemiologists and infectious disease scientists is to just track these numbers, to fill in this part of the table for COVID-19 to see if it skews more to the end of SARS or more like influenza. Now, in order to calculate any of these numbers, of course, you need good data. And a key part of that data is figuring out how many people have been exposed. And that's something that we're really struggling with right now as scientists, is developing the correct test and implementing a widespread testing regime to just assess the spread of this particular virus throughout the population. So I know in the media, there's lots of coverage of the testing protocols that are used. And so what I want to do now is spend a little bit of time talking about different types of tests that can be used and how it's important for tracking outbreaks, particularly for something like COVID-19. The test that I think is most commonly discussed these days, or most commonly being implemented, is one that's based on detecting viral nucleic acid. So the technique used here is called RT-PCR. And basically, it's a very sensitive technique to look for a specific type of nucleotide. You're able to use some molecular trickery to really sort through all the weeds of the nucleic acid that might be in a patient and really fish out just one particular nucleotide sequence that is basically a fingerprint for the virus. And so the procedure is to use a swab to go very deeply inside the nasal cavity to collect secretions inside the patient's nasal cavity. And within those secretions, you will find viral particles. The job of the laboratory technician is to then crack open those viral particles and purify out the nucleic acid genome of the coronavirus. The genome in this case is RNA. And then use this PCR-based technique to amplify a specific region, a specific gene or genes in the genome of the virus. And this is just a pictograph to sort of illustrate what the output looks like in this procedure. If you don't have the virus, there's no signal. If you do have the virus, there's signal. And it's essentially as easy as that. Now, one of the tricks, though, with this technique is, remember, you're only looking for the purified RNA. This says nothing about the ability of that patient to be making infectious virus particles. It simply says that this patient had purified RNA in their nasal cavity. That's it. And that's something that really I want to underscore here, that the test you use is absolutely going to limit what sort of analysis or conclusions you can draw about the spread of an infectious disease. The other major test or type of test that's been getting play in the news media is a serological test to look for the production of antibodies that are specific to COVID-19. And so this cartoon illustrates the kind of techniques that's typically implemented to detect antibodies. Antibodies are molecules that your immune system makes in response to a foreign invader. So when you are infected with a bacterium or a fungus, you name the thing that's not supposed to be in your body, your immune system is going to see that invader and mount a response to get rid of it. And one of the key factors that's made as part of that immune response is antibody production. So these molecules specifically target just that one invader. And so in this case, if a patient is exposed to COVID-19, if they mounted an immune response and made these antibodies, after the infection clears, these antibodies should still be circulating in the patient's blood. And so you can extract a sample of blood and load it in one of these little sample incubation kits. And basically what's going to happen is if you have antibodies present against the virus, those serum antibodies will interact with little pieces of COVID-19 particles that are seated in the sample chamber. All right, and so that's going to form this complex, which basically gets trapped in the sample chamber. And that's when you see the little red line. So if you've seen the commercial for a pregnancy test or used a pregnancy test, you know the idea of when these little lines pop up in the window. It's the same idea, right? Except in this case, we're now detecting the presence of antibodies that would react against this particular virus, COVID-19. All right, and so this test allows you to detect the ability of these antibodies to be produced. But again, it says nothing about what those antibodies really are going to do for the overall health of the patient. So there are issues with these virus testing protocols. And when you read a story in the news media or you're watching CNN, just bear in mind that if you hear that someone has tested positive or you see positive test results from a state or a county or a city, take it with a grain of salt, because you really need to know what the underlying technique was or the underlying strategy was to reach those numbers. So there are a lot of interesting studies that come out, and these are just two that Carol and I pulled recently. So there's a swab test that was detecting just these RNAs from a very small sample of patients in Germany. And what's interesting here is it helps us provide a little bit of perspective about the course of the disease. So after 8 days of isolation, active virus particles were no longer isolated. But again, remember, active virus and viral RNA, those can be two separable things. What's interesting is the presence of the virus is very high in stool samples, which is not something you would typically think to associate with a respiratory pathogen. And I'll touch on this in a bit. That is something that we're now really starting to understand about COVID-19. Its damage to the human host doesn't just stop in the respiratory tract. It does seem like it's moving on to other organ systems. Regarding a serology test, there are some recent studies down in California, one in the Los Angeles area, and I think another one in the San Francisco Bay Area. So there's a few problems with these studies. One of them is perhaps sample bias in the volunteers they got for the studies. But they were able to detect potential exposure to the virus simply by looking for the presence of these antibody molecules. What still remains unclear, though, is are these antibodies any good at generating long-term immunity. And there's reason to come and question that fact, that just because you have antibodies now doesn't mean that they would continue to protect you 3 months, 6 months, 12 months out. And this is problematic, these studies and the ability to really detect how the virus is moving through populations. We really need to know the rate of infection, and it's problematic to generate that type of number when the underlying testing is spotty. And in some cases, the testing might not be applied with the same standard, state to state, city to city, district to district. And this is why you hear many governors pressing the federal government for a unified response. And so there were some studies published in late March, early April, trying to get an idea of what this infectious rate is for COVID-19. Two of the studies highlighted here are PCR tests. The last one is a serological survey. Again, it's not terribly large sample numbers. One group was looking at a homeless shelter in Boston. Another was looking at women in the maternity ward at New York Presbyterian. And this last was one of these serology studies from Southern California. And you can see a disparity here, right? From as low as 2.5% all the way up to 36%. Now, there's a lot you have to unpack here, right? What was the condition of each patient here? What was their underlying medical status? What were their living conditions? Arguably, the high rate from the Boston homeless shelter makes sense because those folks were likely living in a much denser environment than someone who might be living at home in their own home or in their own apartment. So understanding just some of the basic properties of COVID-19 is still sadly elusive. As we're trying to track and understand the outbreak and control the outbreak, there's a lot of effort being spilled into developing drugs or vaccines to defend against this virus or other similar viruses in the future. And so I want to talk a little bit now about some of the strategies to combat diseases like COVID-19. As a basic scientist, I have long been interested in the interactions that microbes have with their hosts. And so most of my work has always centered around the basic biology in the laboratory. And it pains me to say this, but it's true. Very few things that we are able to generate in the laboratory to combat a pathogen actually work out to be useful in human beings. And there are a lot of different reasons for this, but it's certainly the place we always have to start. We always have to establish some of the basic rules, some of the basic parameters of the molecular biology of these infectious organisms before we could possibly scale up to something for, let's say, a drug trial. I found this quote from an immunologist at East Carolina University. One of the things that they say in science is that mice lie and monkeys don't tell the truth. And that's hilarious for a basic scientist to see that. It's absolutely true. Just because you have things that work out in the laboratory does not mean it's going to translate when you move up into a human being. And so that really is why it's so important to carefully conduct human clinical trials to establish the safety and efficacy of any antiviral remedy, whether it's a drug or a vaccine. But that, all those caveats aside, it doesn't mean we shouldn't go after targeting COVID-19. And so what do you target? And I'll throw this slide up. This is back to the basic mechanisms of how viruses like coronaviruses get inside cells. And I just designed your own drug here. I mean, think about the various stages of the virus life cycle. Really, anything is a potential spot for an intervention. And the places that have the big red mark over it now are really the major efforts, or where the major effort is being expended by pharmaceutical companies and government research labs and academic research labs to target coronaviruses. So you can interfere with the ability of the virus to contact host cells. And you can develop a small molecule that inhibits that tight interaction. You could prevent the virus from being taken up by the host cell. Or if you actually get the virus inside and it starts to unpackage, perhaps you could design a molecule that inhibits the ability of the virus to make more of itself. And traditionally, those are the types of interventions that have been the most effective. In fact, there's a drug that's being tested right now. It's made by Gilead. It's called remdesivir. This molecule is probably the one that has the best preclinical data so far that argues that it might be able to at least inhibit the coronavirus from replicating. And this cartoon illustrates how it works. So it's that last red blotch that I showed you from the previous slide. The idea is that remdesivir is going to inhibit the way that the virus is replicated by blocking synthesis of its genome. So it's going to prevent replication of RNA. And you can see here, the molecule it's a small chemical that binds into this key pocket of this enzyme that basically makes this replicating enzyme, this RNA replicating enzyme, it makes it inert. Now, again, it works great in the laboratory, and it remains to be seen how it's going to translate in clinical trials in humans. Regardless of the progress of remdesivir, like any viral infection, treatment for COVID-19 really it's important to support patients with basic supportive therapy. And so it doesn't matter with any type of virus that you see on this list, whether it's going after the upper respiratory tract or the lower respiratory tract. Important things to start with are making sure that the patient has oxygen, making sure that the patient's fever is under control, making sure that they're hydrated. These are all basic remedies for any respiratory infection. And so that supportive therapy is absolutely key. That's why at the outset of this outbreak, the concern was there weren't enough ventilators available for patients who would enter respiratory distress. And that's really where we started with this disease. COVID-19 was billed as being this respiratory pathogen that would destroy your lungs, destroy the capacity of your lungs to exchange oxygen and sustain life, to support respiration. And so what you see in this panel on the left is basically the functionality of a normal, healthy lung. And on the right is when this lung is damaged, whether it's by infection or something mechanical. Basically, the inflammation that results from cells like a neutrophil coming to the site of infection really diminishes the capacity to bring in oxygen into the bloodstream. And it's certainly true. That's a major part of the pathology of patients who are infected with COVID-19. But increasingly, we're coming to understand that it's not just respiratory. There are other organ systems that appear to be damaged by COVID-19. Now, whether or not that's something that's going to correlate with an underlying medical condition of a patient, there's just not enough known at this stage. But just today, there were some news reports out that clotting is now a major concern. That the coronavirus alters the ability of blood to function normally. And so it actually causes it to clot. And this is sort of the opposite of what happens with many other infectious viruses like Ebola. Ebola, it's the opposite problem, right? People infected with Ebola tend to bleed more. And the idea here is just that the virus completely hijacks and subverts the normal homeostasis that keeps a human being alive. So, regardless of where that viral infection starts, maybe it is just in the lungs, but eventually it can spread to other organ systems. So without a cure, what do we do? And I would say the most important things for us to do right now are to just continue what we're doing, prevent spread of this thing, continue social distancing, try and maintain your isolation as best as you can. But eventually, when we start to reemerge from our hibernation, when we start to be out in society again, we need to have a plan. We need to be able to adapt to spikes in the outbreak, which are likely to occur. And so there's a lot of effort that's starting to ramp up, particularly in the New York City area, to develop a plan for contact tracing. So the minute someone is infected with this virus, can we then run down all their close contacts? Can we isolate them? Can we stem the outbreak? Again, providing more time to develop a better therapeutic. And that, of course, is going to require research. There needs to be an intensive effort to come up with a strategy to combat this thing, whether it's developing a vaccine in a timely manner, whether it's getting something like remdesivir to market. There really needs to be an intensive effort at the basic level to get this virus under control. And one of the things I think that's been very inspiring about this entire ordeal is really watching people work together. The scientific community is usually very competitive. Labs can often be cutthroat with each other, trying to get grant funding or trying to get their publications before their competitors. This outbreak has really engendered this new spirit of collaboration, like nothing that I've ever seen. And just watching laboratories work together towards a single goal, getting rid of COVID-19. It's inspiring to watch. All right, that's the end of our presentation. So this is a parting shot, a parting view of our nemesis COVID-19. While a lot of what we talked about today is not all sunshine and rainbows, it's gloom and doom, and a lot of this is scary. I do want everyone to come away with an underlying appreciation of the beauty of the virology, the beauty of the basic properties of microbes like COVID-19. And so this stylized cartoon shows you in really great detail all the macromolecules that come together to make this infectious agent that has basically shut down our planet. The last thing I'd like to say is a little shout out for an upcoming course. So for current Hofstra students who might be watching, during summer session 1, Carol is going to be teaching our virology course. So if you're interested in this course, if you'd like more information, certainly contact Carol. If you have any questions or any interest in talking about this presentation further, you can contact Carol, myself, or Peter Daniel. And you could also do that through the email that you received through the Zoom link. And with that, I'm going to stop my share and take any questions.
Okay, we've got lots of great questions, not surprising. And so I'm going to try to start at the top here. Oops, going the wrong way. It's a long scroll. All right, here we go. So from Ted on 8, does the coronavirus test tell which mutation you got infected with? And if you have one, are you then effectively vaccinated from other mutations?
So my understanding at this point, it's been a little messy with the rollout of the PCR-based tests, and that was one of the issues with CDC. So there are these chemical reagents called primers, and basically the primers sit at either end of a gene that you're going to amplify. And so if you're amplifying a region that is likely to be mutated, then sure, you could detect that change. But I think at this stage of the game, it's mainly about a plus or a minus. Do you have the virus? Do you not have the virus? And then we'll put a pin in it and go back, save a patient sample, and you can do that analysis further.
I also want to point out that coronavirus, this particular virus, has been remarkably stable. There has been a good effort to just constantly sample infected people over time to see, just to look at total genomes in really fine granular detail. And you do see a few variations here and there, as you would expect, but it's been up to now, it hasn't been very long, admittedly, but it's been remarkably stable as a whole.
Yeah, Carol, remind me, what is it? There are 4 or 5 major mutations that keep cropping up.
There's a few deletions that have shown up a couple of times. Again, ORF8. That's the really interesting one, and it's just one of the enzymes or something. I forget exactly what ORF8 does. But there is one that cropped up a couple of times, and actually toward the end of SARS-1, sort of an equivalent one cropped up. So it's really an interesting selection. The selective pressure worked the same way twice.
Okay, next question is from G. Balestas. Is it possible to get an antigenic shift between different types of viruses instead of just viruses of the same type like coronavirus?
Not generally. They don't. They're the proteins that are replicating the viruses, they need certain clues. I mean, they're using their own signals, and there's different signals on different viruses. And so it's like speaking a different language.
Yeah.
So you don't get recombination between
Right.
influenza and rhinoviruses.
Right. So you can't make
They're
there
just,
aren't
but they're
there
talking
aren't
different
hybrid
languages. They
viruses
can't talk to
basically.
each other.
Yeah.
It's
Okay.
not that they couldn't infect the same cell, but they wouldn't recombine.
hmm Okay. So the next question is from Daria Kim, who is coming to Hofstra as a freshman next fall. Good. And uh, sh- she has a question for you, Carol. Why is it that nucleotide inhibitors only work on a specific virus when their RNA sequences are the same?
Nucleoside analogs, well, a number of virus inhibitors are nucleoside analogs. And so they're actually, because remdesivir, right, isn't that right, Nate? It was first developed as an anti-Ebola drug.
Yep.
It wasn't that effective as an anti-Ebola drug, but it turns out it's really good as coronavirus drug. So if I understand the question correctly, the virus, these inhibitors do work against a broad range, and that's pretty much your only target, because a lot of viruses make RNA from RNA, which is something that doesn't really happen in normal cells. That's like your prime target, because you can inhibit that enzyme without inhibiting any cellular enzyme, but it's getting at which kind of decoy nucleotides can fool the virus enzyme without fooling any host enzymes and causing any collateral damage.
Yeah, my understanding was the affinity of remdesivir for the RNA polymerase of the coronaviruses was just higher than it was for Ebola.
Yeah.
I think it's not that it couldn't do it; it just
Right.
that something about the chemistry of that molecule or the shape of the polymerase was not conducive to the inhibition.
Yeah.
Okay, next question is from Claudia. Which is the most efficient and effective test? I guess that's referring to the PCR test and the serological test, right?
That's a really hard question.
Yeah. It depends what you want to know.
Yeah, I think the thing that makes it very, very difficult to answer is the sourcing of all the tests, right? I mean, we're
Which brand
relying on
test?
so many different small companies to roll these things out. After the federal government couldn't match demand, it was sort of like licensing states one by one. I think Northwell is doing a lot of this in New York, and it's unclear to me what the standard is for these various different tests. Um, uh, but again, it's sort of this issue where what are you really looking for? If you're looking for exposure to the virus, I think either test is probably fine. But you can't really do much more than that, right? If you want to know if someone's actively making the virus, well, then you need to culture the virus from the patient. And even with the antibody tests, I mean, it's hard to know what the presence of that antibody really means.
And in terms of which brand, because it's the Wild West out there right now, there's a lot of people putting out these
Mm,
tests, and with it's fine for a short period of time, but ultimately we want some consistency and some quality control so that we know that there's the false positives and the false negatives are minimal.
The beauty of the antibody test is it's something that could be done at home, right? They're designed to be convenient. It's a pinprick of your finger, like for a diabetes blood sugar test. Yeah, smear a little blood on the little sample reader, and that's basically it. The current swab testing, where I mean, if you've seen videos or pictures of what the swabbing is like, they really jam that thing back into your
mhm.
nasal cavity. And to process the sample, you can't do that at home. The instrumentation required is something only a laboratory would have.
Okay, thanks. Next question is from Matthew Friedman. Do you believe that organoids have the potential to serve as better models for vaccine and treatment preclinical trials?
I think,
Certainly. Yeah.
yeah, I think there are at this stage it should be a shotgun approach, right? That we're going to try everything all the time, and you could argue that that's a better model system, um, because I mean it's a way to mimic the human condition in the laboratory, which is better than throwing this into mice or monkeys.
Right. And plain old tissue culture is very, very convenient. I mean, I show tissue culture cell, it's very convenient, but tissue culture cells in so many ways are not like the organism from which they came. And so you always have to proceed with that caveat. Just because something's working in tissue culture, you have to test. And then to animals, of course, as Nate says, we all know that mice lie and monkeys exaggerate or whatever it was.
And those are animals.
Right. Uh, so organoids based on human tissues could be a nice intermediate to give you a better feel for what's really going to work or not work.
Okay. Alyssa Morton wants to know what is happening with the thrombosis effects of COVID.
Good question. I've been hearing about this too, that the clotting, and I don't think it's really clear right now whether it's an indirect effect from an innate response or whether it's, I mean, the you can't isolate virus from the blood. So it's not like there's no viremia with SARS-CoV-2. That is, virus isn't circulating in the blood. So it's not likely to be a really direct effect.
Mm.
But do you know, Nate? I don't know.
No, I mean, I spent a bit of time this morning reading up on this, because it seems like this is the new thing now,
Yeah.
and there are more clinical reports of
With some
patients that,
amputations. Yeah.
yeah, it's really fascinating. I'm gonna try and have it both ways here. I think it's specifically non-specific. I think there is something about this virus that it really, as you said, it really cranks up your immune system to an inappropriate level.
Yeah.
And I think that's the consequence. It's these weird off-target organ systems that are going to pay the price, and for whatever reason, the immune response that's being activated in the case of this infection makes blood clot. I mean, who knew?
Okay, so this is kind of a somewhat related question from Greta Kraus. Can viruses affect one area of the body, but then affect another area of the body years later? For example, I'm wondering about how someone can be at risk for having subacute sclerosing
Yeah. I was just going to use that as an example. Yeah.
encephalitis years after having measles, if they have
Yeah.
measles. I guess shingles is sort of the same thing.
Yes. Yeah.
Mhm.
I don't know that much about SSPE with measles as the kind of much, much later after effect. I'm not terribly knowledgeable about that one. But it's easier to understand with shingles and with varicella zoster because it's a DNA virus, and it's a virus that sort of hangs out and waits in your neurons. RNA viruses haven't generally been known to hang out and wait and last for long periods of time and have a latent period. But there certainly are some examples. And if that's the case for this virus, we won't know for a long time.
Yeah. Lisa Tyne would like to know, is it possible for a virus to mutate into a more virulent strain?
Generally, that's not selective pressure, though. While it's possible, the selective pressure is for as I said, for a virus to be transmitted more easily. So extra virulence generally does not facilitate transmission. Usually reduced virulence facilitates transmission. So selective pressure goes the other way.
Ted on 8 has another question. I have heard along with ventilator shortage, people are concerned about dialysis capacity due to kidney damage. Is that a concern?
Yes, a
Yeah.
big concern. That's again one of these unintended or unanticipated complications. Yeah, that is it's a real concern, and I would expand it even beyond that. There are so many other medical conditions that people have been dealing with uh, at home, right? There are lots of cancer patients who had to delay surgeries because of the way that hospitals have been overwhelmed. So there are shortages that will be really amplified throughout the medical system for months to come. But yeah, the dialysis shortage that's coming.
Now, the kidney damage that we see, it's just secondary to loss of oxygen because that's what because
Mm,
again, there's no virus in the kidneys.
Right.
mm,
That's I just thought that it's really interesting. And of course, as we teach our in Bio 11, how what a high oxygen demand there is in kidneys, it's not terribly surprising that that's going to be the next organ to go if you have a prolonged uh, pneumonia, as these patients do.
okay. Okay. G. Balestas wants to know, will COVID-19 turn into a seasonal virus like the flu, or will it quote unquote go away like MERS?
If I
Well,
if I had
first
to
of
put
all, MERS has not gone away. Lots and lots of camels. Camels are really infected with MERS. Camels got it from bats, and now it's endemic in camels. It's not terribly infectious in humans, which is why there are so few infections of humans. And most of the time, the infected human gets sick and gets better, or sometimes they die, but they don't transmit to other humans. So it's always sort of a one-off every time because the human is mostly a dead end host. So, but then whether it's going to be seasonal, it's hard to say. We don't, the existing coronaviruses that are now circulating in the population 400 years ago, 800 years ago, when they first entered, maybe it was just like this, but we don't know that
Mm,
because, populations are smaller. There's no record of it. I mean, there's lots of sort of random reports of, oh, lots of people died for this. Well, don't know what it was from. It's possible that was like this, and that once everyone is infected or immune, all adults are, and then the only people left to be infected with CoV-2 are children, and children don't have terrible symptoms. And so it may be that 20 years from now, once this has sort of run its course in adults, there's it'll just be another one of those mild coronaviruses. I mean, we don't know, it's a hypothesis, but it's a reasonable hypothesis.
okay. Daria Kim would like to know, what is the reason coronaviruses like COVID-19 cleave their S proteins after host cell attachment?
I can take that.
Yeah, this is
That
for
is
the biologist.
this cleavage of the surface protein is something we see. This is uh, evol- many viruses do exactly that. You see it in HIV, you see it in influenza. It's so often you make a surface protein that has a very little hydrophobic bit, and usually it's the low pH as the virus enters the cell, and an endosome makes it come out, and you need that cleavage. Sometimes the cleavage happens when the virus is on its way out of the cell infected. Sometimes the cleavage happens when it's on its way in. Sometimes it happens in the mucus when it's kind of wandering around in the mucus between cells. But that is just something that has evolved multiple times in viruses. Convergent evolution.
Lisa Tyne, I don't think I got this one before, but maybe, no, I did get this one. Sorry. Okay, so I think there was one more. Oh, I got another question. Just all of a sudden popped up. Ah, okay. This is from Catherine Clifton. With the coming of summer months in the Northern Hemisphere, do you think COVID-19 will decrease in spread with the natural disinfection of increased sunlight? as it does with cold and flu viruses, and increase in the southern hemisphere as they approach fall, winter months. Also, when we in the northern hemisphere get the second wave of this in fall, winter 2020, would it be less infectious due to a more developed herd immunity?
Let's hope that it goes away. I don't think it's going to go away. My hunch is that the seasonal spike that we'll see, or the reemergence of it in the fall, is going to be when we try and be normal again, when schools open up again, when Hofstra students come back, when people start traveling more. I think it's going to have more to do with human behavior than it does with what UV irradiation might do to the viral particles. But who knows, right? I'd love nothing more than for this to all go away over the summertime. But.
I mean, in general, envelope viruses are less stable in high temperature and high humidity. However, I will say seasonality is, with all the opportunity we've had to study seasonality in influenza and colds, it's still not perfectly understood. You see seasonality in the tropics in flu when it's the same all the time. So we don't really understand it. Especially for the first year or so, there's still so many susceptible people, even if the virus is somewhat less likely to survive on a surface because of high temperature and high humidity. The problem is there are so many people who could get it. Then I can't say maybe 4 or 5 years from now.
Yeah.
Yeah,
So,
and realistically, for herd immunity to really do anything for us, you're talking about
we need to have
30 to 60% of the population or more that needs to have had a go of this, and who knows where we're at, right? Based on the metrics, we could still be as low as.
That's why we need testing.
Yeah, that's what I was going to ask. Okay, I've had a couple of people ask, is this going to be made available to the audience? And yes, it's being recorded, and I don't know where it's put up, but do you all know? I know there's a place you can find the recording.
Yeah, Brittany would know.
Yeah,
on
Brittany
the
would know.
YouTube account. Yes.
On the Hofstra YouTube account. There you go. All right, so that answers that question. And I think we're out of questions.
All right.
That's the end of them. All right. So say thanks to the speakers. If you know how to clap, you can clap. There we go. We got a few people. I know where that sneaky little button is. So thanks, and oh, I got some more messages here. Oh, they're all just thank you. There. Okay. All right. So thanks, Carol and Nate, and thanks to the audience for listening. I hope you learned something new.
Yeah.
And stay healthy.
Keep washing your hands.
Yes, indeed. Be calm. Wash your hands. Bye-bye.